So good morning, everyone, and welcome to the FAÇADE webinar series, episode 12. My name is Laetitia Marot. I am a Marie Skłodowska-Curie Fellow at the Honoré Center of Excellence, and today we will talk about biomacromolecules from marine biomass and how to use them in the development of innovative materials. I am very pleased to welcome our speaker, Dr. Susana Fernandes, who is an associate professor in natural polymers at the Université de Pau et des Pays de l'Adour in France. There, she is also the leader of a chair called MANTA that is focusing on marine biological resources valorization and byproducts and marine-inspired materials. Dr. Fernandes is a chemist and material scientist with expertise in functional and bio-inspired materials. Her research areas focus on blue and red biotechnology, blue economy, chemical modification of biopolymers, design of functional polymeric materials from renewable resources, marine biomaterials, and biomimetics. Today, the presentation is recorded, and as usual, you can send the questions in the chat on the right of your screen. I will read them to Dr. Fernandes at the end of her presentation. So now let's talk about biomacromolecules from marine biomass. Dr. Fernandes, you can start.
Okay, thank you, Laetitia, for this nice introduction, and good morning to everyone. So today I will talk about all the research that I have been done, not only here in the University of Pau, because I arrived here just 3 years ago, but I will describe a little bit what I have done. So in different universities, so for instance, at the University of Basque Country in Spain, and also at KTH and Uppsala University in Sweden. So I have divided the presentation in two main parts. First, I will talk about this share, so that calls MANTA, as Laetitia said, that I have created since 3 years almost. And then I will show you some of our research. So the Université de Pau et des Pays d'Adour, it's a university that it's very close to the board with Spain. So we are very close to Spain, and it's in the southwest of France, as you can see here in this map. And it's a small university, so we have two big universities near us, so Université de Bordeaux and Toulouse. So Université de Pau, it's a very small university, but anyway, we are in different cities. So in Pau, also in Anglet, and in Montmarsan and the Tarbes. And so we have 19 research institutions with different scientific research. So, and subjects, for instance, here in my institute at IPREM, we develop research on environment, energy, and materials, but we also have colleagues working with georesources or aquatic environment and resources, etc. So I arrived here at the end of 2018 to create this new research group. It started with a share, and now it's a new research group because we are growing. So we start only with 2 persons, and now we are 12 persons working. So it's on the valorization of marine bioresources and marine inspired materials. So we are in two places. We are here in Pau. We have a laboratory in Pau regarding the part of bio-inspired materials, and all the part regarding the biorefinery and extraction of molecules of interest is in Anglet. So the idea or the general objective of this research group of MANTA is to develop bio-inspired and sustainable biomaterials to lower the impact of the pollution in the ocean. So to do it, we have two approaches. So the first one is to explore, or I don't like this name, but yeah, we we use or we valorize the byproducts or some bioresources from marine organisms. So as a source of new chemicals and new materials and new biomolecules for sure. And then the other strategy is to be bio-inspired by these marine organisms, but also by the way that they live or they are in its synergy between each others. So we have 4 target applications, so biomedical, cosmetic, packaging, and nanocomposites. But for instance, now we are also starting a new collaboration with colleagues here at UPPA on materials for construction. So the group is divided, or our research subjects are divided in 4 for the moment. So first is this big one. It's the valorization of the marine bioresources and fishing byproducts. So we have some fishing industries here in the Basque Country coast that aims with us to valorize their byproducts and wastes. So we do it by the extraction of biomolecules of interest. And then, as I said, we do materials. So different kind of materials, hydrogels, films, 3D scaffolds, composites, etc. And to close the loop, actually, we also are interested to see the impact of these materials on the human health. So because we do some materials for regenerative medicine, but also for cell protection. And the other is a new field for me. We work in collaboration with some colleagues here at IPREM. It's to study the impact of our materials in the marine environment. So this is just an overview of all the people that work in the different subject, in the different research topics or areas. And this year, so we have the pleasure to welcome a biochemist and enzymologist. So we were awarded with another big project with a junior share. So because I was missing, because as Leticia said, I'm a chemist of materials, so and to do green chemistry and we were missing someone in the group with an expertise in enzymology. So is Zhang. She started in August to work with us. So we work. So she's working at the interface of all these research topics. And as I said, so we are exploiting these marine bioresources, but we don't want to do like we did with the fossil resources. We want to do it, but through bioethic values, and also respecting the acceptability of the society regarding our new materials and the way that we are exploiting these bioresources. So we have, for instance, a collaboration with CBIOS, that is the European Center of Expertise in biomimetism or biomimetics. And we are working, we have 2 students working on this, a sociologist and an economist working on these topics. So now let's move for the second part, that now you know a little bit about my research. I will show you some research that I have been done, but as I said, not only here, but in the other institutions where I worked before. So I will first present some works that we have done from using marine bioresources, so the extraction or valorization, and then the development of some materials. So here in this slide, I have, for instance, some of the marine organisms that we are using to valorize the bioproducts, and we will be focused in the first part, or in the main part of of the presentation of chitin and nanochitin from crab and shrimp, and then I will show you a material that we are developing by using mycosporins that I will explain you what is. It's a bioactive molecule that we extract from the local red algae. So let's start by talking about how we used or how we valorize the wastes from the exoskeleton of the crustacea. So chitin is the main biopolymer that we can find in the exoskeleton of the crabs and shrimps. It's a linear polysaccharide, and it's very similar in nature, has the same function as cellulose, so it's a structural biopolymer. But in carbon 2, instead of to have a OH group, we have its acetylated polysaccharide. So chitin, it's a fantastic polymer because it's biodegradable, it's biocompatible, but during a long time it was considered like an intractable polymer because it's very difficult to dissolve. And this, if you work with materials, you know, if you have something that you can't dissolve, it's difficult to do a material with it. So, but during long time, we valorize chitin in chitosan. So chitosan is, we can have the chitosan just with this, deacetylation. We deacetylate this group and we have amino groups. So then we can dissolve this polymer in the form of a chitosan, and we can has the same properties than chitin, like biocompatibility and biodegradation, but has these properties, the film forming ability. So we can do a solution and then we can manipulate it in order to have different materials. But since most than 25 years ago, so we start to regard to chitin in the pristine form. So because in nature, as I said, it's like cellulose, so we have it in a fibrillar form. So here you have a schema of how it's composed, the exoskeleton of here it's a lobster. So it's the chitin chains. They assemble together to form these nanofibers, and with different techniques, so or using chemical treatments with acid hydrolysis or some physical treatments like grinding or microwaves or high pressure homogenizer, we can, or to have chitin nanofibers or chitin fibers and chitin nanocrystals. So we worked, I started working on it when I was in San Sebastian in the Basque country with Asier. So, and we in the group of Jalel Labidi. So we developed a way to extract chitin nanofibers. And as you can see here in this IFM image, they were very long, with a very high aspect ratio, what is very important for us in order to do materials, composite materials. So we also want to see if these materials has antimicrobial properties. So we did some fungal activity, and we show, as you can see here in this graphic, that both forms, chitin nanocrystals and chitin nanofibers, have better antifungal properties than the chitin in the powder form. So this, it was good to have this kind of properties. And we also developed with Ruth Fernandes. She did a great job using microwaves assisted extraction, using different wastes or byproducts from lobster, shrimp, and squid pen, and to try to have nanocrystals and nanofibers from these different species. And we found that using an acid hydrolysis, but with microwave, combined with microwave, we were able to have the chitin nanofibers or crystals, and we were able to reduce the time of the isolation of these nanocrystals and also the concentration of the acid. So this, it was also very good work. So why we were interested to have this different kind of nanoforms of chitin it was to do materials, so for packaging and for tissue regeneration. And we were interested to see what were the differences between using chitin nanocrystals or chitin nanofibers in these materials. So now I will show you some of these materials. We did a lot of materials doing this study, so to see the differences between using chitin nanocrystals or chitin nanofibers. And we used, for instance, we did nanocomposites using starch, chitosan, PLA, and cellulose as matrix. We did different kind of materials, as you can see here. For instance, bag plastics. It was a big European project that we developed at the Basque Country University. We also did this kind of composites, and using melt mixing and injection process. And we also did some freeze drying process in order to have 3D porous scaffolds. And for sure, we characterized all these materials. So I choose two of them to show you today. So actually, we used starch as matrix, and we used, I will show you the differences between the materials prepared by casting evaporation method and using the melt mixing procedure. So we again, here it's I start with the materials processed by casting evaporation method, and we also were interested because we were thinking about these materials for packaging. So we see the inhibition of the growing of the fungi at the surface of the films. And for instance, here in this first image, we have the starch film, and as you can see, it's after 7 days of incubation, they are almost covered with the fungi, and we see that has a little difference between the films that were that we incorporate nanocrystals and nanofibers. So as we can see here, we have a little bit better results with the chitin nanofibers, because we think that there are the groups the amino groups, they are more available in this kind of materials than with the nanocrystals. That we have with the nanocrystals only the crystalline part, and here we have crystalline and amorphous part, and we think that it's because of this. So then we also compared the mechanical properties, and as you can see here by the Young modulus, so here it's starch, our control, and we have better mechanical properties using the fibers, the nanofibers. This is because we have the percolation effect of these fibers in these materials, and not that was not verified with the chitin nanocrystals. If we compare these mechanical properties with the same, using the same quantities or different amounts of chitin nanocrystals and nanofibers, but using the other process, so the melting mixing process, we have exactly the same observation. So we saw that with the chitin nanofibers, again, due it to the percolation effect, so we have better mechanical properties using nanofibers than chitin nanocrystals. So this is our conclusion. So we saw that actually the amount of these nanoforms in the matrix and the morphology of the nanoforms, they have these different impacts on the final properties of the material. So now I will show you another material that we prepared. So it was done in collaboration with the Faculty of Medicine in Bilbao. Actually, we prepared the 3D scaffolds that I presented you before. And the idea here, it was to see if our biomaterials were able to differentiate to cartilage. So how we did it? So in this time, we didn't use starch as matrix, but we did materials with chitosan as matrices. And here we have the schema. So again, we used chitin nanocrystals and chitin nanoforms to see the differences for the cell adhesion and proliferation, if these morphologies have an effect. So we prepared 3D porous scaffolds, as you can see here, and also 2D films that were very flexible, as you can see here in this photo, using chitosan, as I said, and ginipin, that it's a natural cross-linker as cross-linker. So what we verified, that all the materials were not cytotoxic, so we can use all the materials for to grow or to do the cultures with the cells. And then here we have some images with SEM, and we can see how. So we did the live the life dead test to see if the cells are the cells survival and grow in this kind of materials. And as you can see here in green, it's the cells that we have just some points in red that are the dead cells. So the major, it's the cells that are very happy. And we can also see here that these are the three, so here it's the films and here it's the 3D scaffolds. And we can see here that the cells are completed there in the cell wall of the walls of our scaffold, and they are adherent and proliferating. What it was very good to do our materials. And here at the surface, you can also see the nanocrystals at the surface of our films. So what we also see with this, with these materials, it was that actually here we had better results using chitin nanocrystals because the rigidity of our films, our materials increased. So we used these materials to create our new or to do new scaffolds. And in this time, to do the differentiation, we used something more real. So we didn't use individual cells, but we prepared spheroids with stem cells to do, and we used hypoxic conditions in order to have this cell differentiation. And we have demonstrated it with a significant increase of sulfate glycoaminoglycan synthesis that we prove it in our materials. So now these are, it's just some reviews that we did. We were invited to do about our nanochitin work because we have very considered a lot of work on it. So we have been invited to do some review articles. And now I just, before to finish my presentation, I will just show you a material that we have been developed since some years now, I think 4 years. Actually, I don't know if you know how, for instance, this was a question that I have one day, how the fish, they protect themselves from the UV radiation. So, and I did some research about it, and I found some molecules called mycosporins-like amino acids. And for instance, the fish that they live in the reef, the corals, you know, it's not deep at all, the water there. And actually the fish, it's not like us. If we wanted to protect ourselves from the UV, so we can be inside, or we can put some clothes or to buy a sunscreen. But the fish, it's not like, so like us, they can't biosynthesize this kind of molecules to protect themselves from the UV radiation. But actually they eat some algae that they synthesized to protect themselves from the UV. these molecules that they are secondary metabolites that cause mycospores and mycospores like amino acids. And we don't know for the moment how it happens, but the mechanism, but they eat it and then they have these molecules. We can find it in the mucus, at the surface of their mucus and also in their eyes. So as a materials chemist, scientist, as I work with this kind of materials, so hydrogels and this kind of materials, I was thinking, wow, we can actually to extract these molecules from the algae and then to incorporate it in this molecule. So it's what in the hydrogels. So it's what we have done. So it's very cool, these molecules. Here I have an example. So for us as chemists, it's fantastic. So the molecule has absorption capacity very high, even higher than the molecules, the synthetic molecules that we can find in our sunscreens. Also present antioxidant activity. There are some works that show that they are activators of cell proliferation. And me as a chemist, we also have the functional group set, so we can use it to link these molecules to other molecules and to materials, because for instance in the algae they are in their free or state, but in the mucus of the fish they are linked to polysaccharides or to proteins. So we are trying to mimic this kind of materials. So we did the first work by grafting these molecules to chitosan, and we show that this material, so we were able to do a film, and we showed that these materials were absorbing the UV, and these materials are also biocompatible, and they are not cytotoxic. And now we are trying to prove that they really will protect us from the UV radiation. So we are not the only people that are working with these molecules doing materials. So there are some colleagues from the King's College of London, but they are really mimic like the fish. So they are doing some pills. So this is some cartoons that I found in the net about these two works, so that some people are joking with our work. So here it's how they see the work that the colleagues of King's College of London they are doing. So actually, as I said, they are trying to mimic what the fish do. So they are doing some pills and just to take like this. So I know that some years ago they tried to do some tests with murine, but these molecules were not found at the surface of the skin of the mouse. And yeah, but I don't know what if they still working on it. I think that they are trying to do something to protect these pills, like a coating. Maybe it's in our stomach that the pH is very acid. And this is also a cartoon that I see about the work that we have published it, that is someone that is going to the beach with the fish. But I assure you, if we have one day some materials that will be able to protect us from the UV, it's not like this that I'm expecting that we go to the beach. So before I finish, I would like to thank my team and all the institutions and all the people that I have working. Yes, and the funding. And thank you, Leticia, for sure for this invitation, and thank you to all of participants.
Yeah, thank you so much. Very, very, very interesting presentations and cartoons. I have a question, because at InnoRenew we are working mainly on the building environment, And at the beginning of your presentation, you mentioned that you are also starting to work on material for construction. So I would like to ask you what type of material do you consider, and if you can tell us more about it.
Yeah, I can't tell you a lot of things because we just submit an European project, but yes, it's with the concrete. Yeah, because there are a group here at university that they are specialized in concrete, and we are proposing to do bio-based concrete.
Okay.
It's on it. Yes, it's in concrete.
Super.
Yeah.
We have a question. So also participants, please don't hesitate to post your questions in the chat. Now we have one question from Václav Sebera. Thank you for your interesting talk. He has actually two questions. Is it possible to synthesize, obtain long fibers from chitin, for example, for mechanical purposes? And can chitin be used for making adhesives?
Good question. Thank you for your comments and questions. So long fibers, it depends of the, with the homogenizer, we were able to have fibers with some microns, 1 micron of long and around 100 nanometers of the diameter. So I don't know if is this the question. And then, yeah, it's so what, maybe you want fibers, not at the nanoscale. I don't know if is this that you are asking. We, for instance, we are trying to have the fibers, not at the nanoscale, but yes, fibers with chitin. For the moment, we don't have the dimensions that we would like, but we are working on it using mechanical assisted techniques. And the other, it's for adhesives. I don't know. I don't know. Chitosan, I know that there are some works that are have been done with using chitosan. Maybe because maybe have something between, because maybe you want to have the mechanical properties that are inherent to chitin, and when we are going to the amorphous part of chitosan, we are losing these mechanical properties, but maybe something between. And I know that it's possible. I have some colleagues that are trying to work on it. So chitin, I don't know if you can use as an adhesive, chitosan for sure. But why not to find middle, something in the middle?
Okay, thank you very much. Thank you again. We don't have more questions, so I think we will conclude here. Thank you so much for the presentation, for answering the questions. Thank you to all the participants who joined us today. And I hope you enjoyed the webinar, and I hope you will come back to see one of the future webinars. So bye and have a good day.
Bye-bye. Thank you.